Applications of recombinant DNA technology. Now we are going to conclude our topic or the subject, that is gene manipulation and genetic engineering, because we have discussed most of the topics that are related to the study of DNA, how it can be exploited, how it can be used to transform bacterial, plant or animal cells. And now we are going to focus that what benefits we can get from this technology of the genetic engineering or recombinant DNA. And here we are first going to focus that different ways to exploit recombinant DNA technology. Starting from the recorded history of the human, we have the idea that plants or animals, they have been used for food, for fiber, to get different type of chemicals, dyes, and even antimicrobial drugs to cure different type of infections. But now we have the technology by which we can improve or modify such traits. अगर हम ये देखें कि 1980s तक हमारे पास ज़्यादातर जो प्रोडक्ट्स थे वो हम हासिल करते थे by exploiting the natural diversity of the living organisms, कि जो नेचुरली हमारे पास चीज़ें अवेलेबल थीं, चाहे वो प्लांट से या एनिमल से या माइक्रोब से, हम वही एक्सप्लॉइट करते थे अपने बेनिफिट्स के लिए. Or even up to 1980s, if some traits were improved, they were based on mutagenesis or breeding. कि हम plant breeding की या animal breeding की techniques use करते थे या mutations create करवाते थे animal या plants में to get our beneficial products. But recombinant DNA technology have revolutionized that how we can exploit animals or plants for our own benefits, because now we have the idea how to transfer foreign DNA or how to alter the endogenous DNA so that one can get novel strains, novel varieties of plants or even animals that can perform some specific task. If you remember, the first human protein that was, you can say that, produced by using recombinant DNA technology, it was insulin. So when gene of insulin, it was transferred into the host organism like E. coli, it results in expression. Then after this insulin, hundreds of human proteins that were very important or vital, they were synthesized or produced by using this technology. Now we have the idea that how we can alter the metabolic pathway of the organism, कि जो कुछ chemical reaction cell के अंदर चल रहे हैं उसको किस तरह से alter या modify किया जा सकता है by altering the DNA of the host organism. It is not only the proteins that are important, rather some phenotypic markers, they are also very valuable. For example, in case of plants, we need some important varieties that are able to tolerate pest attack, that can resist against different type of fungal or bacterial diseases, and even if they are able to tolerate environmental stresses, and they are equally important. So such type of traits to tolerate environmental stresses, they can be introduced by using recombinant DNA technology. It is because of the use of this advanced technology that millions of hectares of land around the world, it is under cultivation by using transgenic plants. Now, This technology, it is not limited to only plants, even animals, they have been transformed, as we have already discussed. And the concept of gene medicine was introduced, where some lethal diseases like cancer, it can be treated by using some sort of foreign genes that can repair or that can kill the cancerous growth. So these are the different ways by which we can exploit the recombinant DNA technology, that we can improve the microbial traits, plant traits, animal traits, and finally we can get valuable end products, mainly in the form of proteins or hormones.